Okay. Good afternoon. Welcome to this IMC Distinguished Lecture. So as you know, we have been running this series as part of the Azadi Ka Amrit Mahotsav. Today it's immense pleasure to introduce today's speaker for me. It is Professor Vinay Nandicoori. Vinay happens to be my collaborator. We collaborate a bit, and so it's an absolute pleasure to have him today. I won't take much time introducing Vinay. He needs actually no introduction. Professor Vinay Nandicoori happens to be currently the director of the CSIR Center for Cellular and Molecular Biology, CCMB in Hyderabad. A bit about Vinay. Primarily as an independent researcher, he has been working on delineating the signaling networks of Mycobacterium tuberculosis for now 15 to 20 years. Vinay did his master's at IIT Bombay, subsequently his PhD in ISC Bangalore in the MCB department. He did postdoctoral work at Texas A&M University and University of Virginia. In 2004, he returned to India and started his independent group at NII. And for slightly less than 2 years, he has been the director of CCMB. He has a lot of recognition. I don't have time to go into that. For example, he is the fellow of all 3 academies of India. He's a J.C. Bosch National Fellow. He has got the National Bioscience Award for Career Development and the NACI Scopus Young Scientist Award. There are many more. I won't stand between you and Vinay. So Vinay will be speaking today about delineating survival strategies employed by M. tuberculosis. As you know, March 24 happened to be the World TB Day as well, and this talk is happening at the right time. So thank you, Vinay, for accepting our invitation and looking forward to your talk.
Ajit. Thank you. So I know this is not the audience which I generally give talks in, so please stop me if I'm really not making any sense or talking about things which are not exactly in your comfort zone. I would rather give us brief, curtailed talk than talk to myself, rather. Any talk that I start with my group, actually. The reason is that can you shut off these lights? That one too. No, this big one. You're shutting everything other than the big one. Not this. This light, which is the big light. Now you can put on all the other lights, otherwise everybody will sleep. Okay, so any talk that I generally start, I tend to start with my group. This is my current group at CCMB. Most of my lab has moved from National Institute of Immunology to CCMB in the past 1 year. Most of the work that I am going to present is done by a girl named Mahek Zahoor Khan here. That is what I'm going to talk about today. TB, for all of you, is basically a very old pathogen. You go to Egyptian mummies, you'll find it. You would find it in different forms. It's called different names over the period of centuries, and in fact this is thousands of years old. TB is basically called various names. You can see some of them. I really don't want to read it out. But at some point of time, it was even considered fashionable to get TB because you become very pale. So, so somehow the thing is considered okay when, paleness is considered fashionable at some point of time. But anyway, eventually, Robert Koch has figured out what is responsible for TB. So he is a German bacteriologist who discovered not only the bacteria that causes TB, but also that which causes anthrax, septicemia, and cholera in addition to this. He is the one who came up with these postulates, which is important, for an organism is causing a disease if the organism is present in every case where there is such a disease is found. It can be TB or cholera. The organism is found in every case. And you should be able to culture the organism outside, right? You take it out and you should be able to culture it. And finally, if you take that culture and put it back into an animal, it should cause disease. And you should be able to get it out again from that animal system. So this is what he would, in those days, considered as Koch postulates, and these are a really important set of things that are that defined what is a pathogenic bacteria. And this is even today valid, but it is not going to work out always because some of them are very difficult to culture, like leprae. But even they can be cultured, but they need a special niche for culturing. So Mycobacterium tuberculosis, all of you know, is basically Mycobacterium tuberculosis is something that basically it causes disease. Dheeraj, can you do it? So Mycobacterium tuberculosis mostly causes disease in our lung. Whenever you're traveling or any other place or wherever, people cough. That is similar to what happens in the case of SARS-CoV-2. So this transmits the aerosolic droplets from one person to the other person, and it gets into our body, eventually causing a respiratory disease, which is basically it causes disease in lung, right? And despite all the things, it is still a major killer. 1.5 million people died in 2020 alone. India still has a very high burden of tuberculosis. And the bigger problem is, as it is true with many bacterial diseases, you have multi-drug resistant bacteria that are emerging because of misuse of drugs and many other reasons. One, of course, organisms tend to evolve. We think tuberculosis to be a disease that comes in the lungs, but 15% of the cases, tuberculosis is found in other organelles, including brain, lymph nodes, bone, stomach, even in reproductive organs, and eye, ocular TB. So it is found in many other parts of your body, and these make it difficult. In fact, Egyptian mummies, they are basically recognized that they have TB based on their bone TB. Bone TB makes it basically bent and completely, you find granulomas there. So things like that basically tells you that this pathogen can move around within your body. Even if you clear it from the lungs, it doesn't mean that it's completely gone. It gets into something called latent infection. So what happens when you actually encounter a person who has tuberculosis and his aerosolic droplets have entered your body? They are taken up. All of you would know that we have two levels of immunity: innate immunity, which is essentially macrophages, dendritic cells, and other things. And then you have adaptive immunity, which is basically B cells and T cells, which produce antibodies, and T cells directly kill the organism. So what Mycobacterium tuberculosis does, or it gets taken up by immune cells, which are like macrophages. Unlike many pathogens, macrophages generally are supposed to take it up and clear the pathogen. Unlike many other pathogens, it does not happen in the case of TB, because tuberculosis, Mycobacterium tuberculosis has an exceptional ability to survive within phagosomal compartment and does not allow its own degradation within the innate immune cells. So it basically, instead, host comes up with an alternate mechanism of trying to wall it off in a granuloma kind of a thing by surrounding it with many other immune cells, including neutrophils and other lymphocytes. You can see that. That's called basically a granuloma. And this granuloma, at that point of time, has three different fates it can go through. If you are amazingly good, your immune system is like too robust, it can actually completely clear it, and in which case it's called calcified granuloma. Still there will be a damage which you can visualize it, but it's calcified granuloma. And if your immune system is not good and you are nutritionally deprived or you are immunocompromised for any particular reason, disease can be established. When the disease is established, it starts moving to other parts of the lung, or it can go into any other niche that is available for it. In many cases, however, we come to a point where granuloma is kind of what we call as latent, in which pathogen is there, it's not dead, but at the same time it is not active disease. When the conditions are right, it can be many reasons including age related, it can actually become activated, and when it gets activated you will end up getting disease. So for example, quite a lot of people who get extrapulmonary tuberculosis, they realistically may not even have anything in their lung, but they end up getting ocular TB, they end up getting gastrointestinal TB, you end up getting TB in other organelles. Sometimes it is along with both, sometimes it is solo. Yeah. Yeah, very interesting question. In fact, this is a question that we would like to address. Here is the problem. Most of the extrapulmonary tuberculosis, you don't get enough samples to directly take it for sequencing. And if you have to culture it, you don't know if you even maintain the heterogeneity, specifically gene expression pattern, correct? Because the niche has changed. So only way this can be addressed is if we actually can get enough tuberculosis cells. I mean, it is most probably possible in case of bone TB to address this question, but in case of ocular TB or gastro TB, intestinal TB, or sometimes skin TB and lymph nodes, some of these things we can address this, but we have to make a single cell transcriptome kind of an experiment to address this question. Anything less would not work. So these are some of the new techniques that are available, but there is not much research on this yet. But I personally believe the profile is likely to be different. Right? So why worry about TB in a COVID era? COVID has killed, this is a little bit outdated number, let's consider it around 7 million deaths. And lots of cases over the last 3 years. But the reality happens that 7 million is roughly, TB can cover that in about 4 to 5 years. So it continues to be the problem because COVID is not really killing that many people because many of us developed some level of herd immunity or what you call as an hybrid immunity. Because of that, we are not getting huge levels of death in case of COVID. But TB is still killing and it continues, it will reach the same numbers in about 4 or 5 years. My lab, we started working on tuberculosis when I came back to India. Prior to that, I wasn't working on tuberculosis either. So I was basically a signaling, mammalian signaling, working on prostate cancer before I came to India, and then I kind of decided to work on tuberculosis and signaling. So that's my, basically most of the work was, and rest of the areas kind of came out of that because you talk about a signaling, there is a kinase, there is a substrate. So we started looking at substrates. That's how I went into peptidoglycan synthesis, cell division, and at a later point of time into even transcription. So today I'm going to focus on essentially transcription, and I'll give you a bit of essentially my first law, which is signaling. What is signaling from the context of anybody who is basically looking at any cell? Something, you get an extracellular stimuli and you have a cellular response. So this is equivalent to a relay run, except that in each position, the relay, you instead of giving it to one person, you're giving it to 4 people, and the 4 people give to 4 people, 4 people give to 4 people. By the time relay happens, you get an amplified signaling. That is what happens in mammalian cells. And there are tyrosine kinases, there are serine threonine kinases, right? What is a serine threonine or a tyrosine kinase? Is simply the matter that the hydroxyl groups on a tyrosine, serine or a threonine are phosphorylated by the kinase. The phosphate comes from ATP. Now you have a tyrosine or a serine or a threonine which is phosphorylated at the hydroxyl group on the side chain, and you need a phosphodicate to take it off. So what happens when you have a phosphorylation? The local conformation changes, sometimes changing the activity of the protein, sometimes changing the localization of the protein, sometimes changing its interacting partners or immediate vicinity. So you can change the functionality of a protein, immediately having a biological consequence. That's how phosphorylation happens. In case of bacteria, it is well known till about 1990s that basically bacteria is thought to have a different signaling mechanism called two-component signaling mechanism. In this, what you have is a receptor histidine kinase, not serine, threonine or tyrosine, histidine kinase. Histidine takes up the autophosphorylates and gets phosphorylated. From there, the phosphate is transferred to its response regulator on aspartic acid residue side chain. So eventually that changes the transcriptional activation profile of the response regulator, making it a transcription factor. Basically it goes and binds to, along with the sigma factors and otherwise also, to promoters, thereby giving you a bunch of activations. So this is equivalent to a system where you have one kinase, one response regulator. They are cognate, they are pairs. And however, in 1990s, serine threonine protein kinases were started appearing on the scene for bacteria too. They are like very eukaryotic like. Specifically, any serine threonine kinase generally has two structures. It's like a snowman. I keep on saying this, it's like a snowman. It is the upper part of the snowman and the bottom part of the snowman, a little bit fatter. This is full of alpha helices. This is full of alpha helices. The N-terminal region is full of beta sheets other than one helix. ATP binds here, and the phosphorylation happens in this region. So this is how kinases work, right? And Mycobacterium kinases are there, then you need a phosphatase to take it off. In Mycobacterium tuberculosis, there are 11 two-component signalings and 11 serine threonine protein kinases, 1 phosphatase and 2 tyrosine phosphatases. So 11 serine threonine kinase, 11 two-component signals are anyway important for the Mycobacterium tuberculosis, but it seems to have distributed its work. The reason I'm saying is in E. coli, which is much it's a different organism altogether, and it has approximately 60 two-component signalings. So if you consider that Mycobacterium tuberculosis actually only has 11, so we consider it is distributed its work among two different systems. So we started working on it. How does a kinase structure look like? Kinases in case of Mycobacterium tuberculosis have intracellular kinase domain with a small transmembrane domain and a small extracellular domain. So it is equivalent to extracellular domain takes up the ligand and it gives you the biological response. Over a period of time, in the last 14-15 years, or it's almost 17 years, we have worked on 3 kinases. 2 of them are named PKNA, PKNB and PKNG. Protein kinase A is essentially a kinase which has a very small extracellular region, you can see that, but it has a transmembrane. It is also holding onto the membrane. PKNB, on the other hand, has a very prominent extracellular domain, and we will tell you what it works on in a while. PKNG is the only soluble kinase, and it is also secretory in nature. So PKNA and PKNB in the kinase words of MTB are essentially those which are essential for bacterial survival, means you cannot knock them out. Mycobacterium tuberculosis encodes for 4,000 genes, out of which 800 of them are essential for the bacterial survival, with 3,200 being dispensable for bacterial survival when you grow in outside media. Right? So what we did over a period of time is we done enough work, and basically I'll make it very, very brief here. I'm not going to go heavily into it. Okay, how do you analyze the function of a protein? You overexpress or you take it out. When we overexpress, what happens? This is not done by us. This is done by a guy from Harvard. When they overexpress, what they found, whether a PKNA is overexpressed or a PKNB is overexpressed, you can see that the cell shape changes. It becomes elongated or it becomes bulgy. And so this was way back in 2005. They proposed that protein kinase A and protein kinase B are involved in the cell division. Okay, so what we did is we generated what we call as a conditional knockouts. What's a conditional knockout? Knockout is something you take out the gene. Conditional knockout is you keep the gene, but when you add a particular condition, it can be anhydrotetracycline, XYZ, anything, you turn down the gene, like you stop expression of the gene. That's called conditional knockout. Using that, we showed that both pKNA and pKNB are essential at every phase. Means you cannot take it out whether it is in vitro or when you infect cell lines or when you infect animals. In all these cases, you take it out and you don't get any colonies. This is something we showed, right? But we were still perplexed that how the signaling happens. Protein kinase A did not have a very distinct extracellular region, so we did not work much on that. Protein kinase B, on the other hand, had a very distinct extracellular domain, and work from other groups showed that this extracellular domain binds to peptidoglycan fragments called neuropeptides. So I know that most of you are like, basically peptidoglycan is made of two sugars, N-acetyl glucosamine and N-acetyl muramic acid, with 4 amino acids attached to N-acetyl muramic acid. So this is a chain of that is what peptidoglycan is. And the cross-linking happens because of the amino acids. So you have linear chains connected through cross-linking of the amino acids, a meshwork right outside the membrane. So what we showed is that if we have identified the residues, we mutate the residues that are involved in the interaction with the ligand, protein kinase B localization changes, it becomes hyperactive and killing the cell because of that. Means the not having pKNB or having an improperly localized pKNB or a overexpressed pKNB, all of them can kill the bacterial cell. So why that basically means it can be a good drug target, and we are still working on the possibility of actually targeting protein kinase B for therapeutic intervention in the context of tuberculosis. Protein kinase G, on the other hand, at that point of time, there was a paper published in Science which said that I take out pKNG. I told you a while ago that the bacteria survives quite well in phagosomal compartment. If you take out pKNG, it gets degraded by fusing with the lysosomal compartment, thereby completely getting degraded. So this was important for survival in the host. What we showed is that pKNG seems to be controlling metabolism in the pathogen, and this metabolism regulation seems to be important for its survival in low oxygen condition or nutrient limiting condition. In fact, we went on to show that if you don't have pKNG, in fact you do not have reactivation of the disease. We have done this experiment in animals and we showed that you don't have a reactivation of the disease. When does reactivation happen? Remember, initially I showed that it goes into a latency. If you can eliminate the latent bacteria, you will most probably, or you will not have any reactivation. So if we can target pKNG in addition to a replicating bacteria, the chances are that we will eliminate latent bacteria. A hypothesis that needs to be tested by developing certain therapeutic molecules, certain molecules that can be used as inhibitors. We have not done it yet. So we were working on this. At that point of time, my student came to me and told that there is this how do we pKNG gets regulated or whatever. She wanted to look into transcriptional regulation. She came up with some hypothesis and basically said that there is a particular transcription factor that works on it. This is the girl who has done all the work. Her name is Mehak, and you have Biplab is a student of mine, and this is done in collaboration with Gaurinath and Shrikant. So I'm now shifting the gears from kinases to transcription. Transcription generally is, yeah, please. Yes. And rather absence makes it worse. Presence, it stays comfortably there. So in the absence of pKNG, you have higher levels of fusion with the lysosomal compartment. So at that point of time, it is thought that PKNG is secreted and controls certain things. And even now, that particular thought process is pretty much alive in the field. A lot of people believe, there is a 50% of the people believe that PKNG is secreted, and it is shown to be secreted. And this secreted PKNG is interacting with some of this machinery that is involved in the fusion, RAB machinery. But I believe it is compromising the pathogen itself in the nutrient limiting condition, because we have shown that the metabolism changes and that in itself compromises the pathogen, thereby fusion happens better. So both are possible, or maybe both are contributing together. Whichever way, PKNG seems to be an important molecule in inhibiting the phagolysosomal fusion.
Can it be checked by generating some domain is necessary? If some domain is specifically necessary to, let's say, bind
Kinase
to the
activity is necessary.
no. So if it's secreted and it's binding to the RAB proteins, it may not be necessary that it's the kinase domain going and binding. So any other domain?
So in any other domain, no. It's still the N-terminal region and the kinase domain seems to be important. But even if it is getting out and it is interacting with the RAB, doesn't mean that the kinase activity is not involved. So that part is still very fuzzy. People have shown it interacts and it does this and it does that. Papers are published both ends. I am more of the first school of thought. I believe that the pathogen itself is compromised. Secretion is a smaller part of the story. That's my belief. But if I give it anywhere, there is an argument on it. So basically, transcription. Okay. Now, out of the 4,000 proteins that are hanging around in tuberculosis, 200 of them are transcription factors. So what's the transcription factor? Transcription factor interacts with, if you know, the bacterial transcription is driven by sigma factors and RNA polymerase. Sigma factors are required for recognizing the promoter, and then they recruit RNA polymerase to the promoter. But that is not the only regulation. Quite a lot of transcription factors change the affinity of these complexes to the promoters. So transcription factors essentially form a complex, sometimes activating complex, sometimes repressing complex. It's not necessarily always activator, right? What we were looking for is a transcription factor that is not essential for bacterial growth when you put it in the culture, in vitro culture, in the sense when you add media and all. But it is essential for survival when it is in an animal. This data was already available. All that we had to do was mine it. And we got 10 such transcription factors which we can knock out outside, nothing happens, but you put it into a host, they will have a phenotype based on the data. And out of which only one of them was very unique to actinomycetes, which is very strange because, only one transcription factor seemed to be unique. So we wanted to work on it. We basically went and looked into the opronic arrangement of this particular transcription factor. You guys know that bacteria, it's all oprons, not a single gene, multi-genes. So the second gene in this opron is RV1332, is a transcription factor. And it is a transcription factor which is very small, 218 amino acids. It is non-essential, and it is essential when you actually put it into a cell line infection or a mice infection. It is unique to actinomycetes. Like any good geneticist would do, what we did, we generated a knockout. Forward genetics approach, you generate a knockout where you replace the gene with an antibiotic marker so that you have a way to select for the mutant. Let me tell you, tuberculosis grows. How many, I don't know, this is a different crowd, but in E. coli colony comes in about 12 hours. And E. coli colony, E. coli divides every 20 minutes to 25 minutes. Mycobacterium divides once in 20 hours. You may be wondering why the treatment is also 6 months, right? This is one of the main reasons. It doesn't divide fast enough. It divides as slow as our cells. And sometimes if the environment is not good enough and oxygen is deprived, even slower. So if you are looking at chasing a knockout in Mycobacterium tuberculosis, it is, if you are successful, 2-month work. If you fail anywhere along the way, you're talking about months and months of work. it is a relatively challenging problem to create knockouts in Mycobacterium tuberculosis for multiple reasons, including a terrible homologous recombination, very bad transformation efficiency. All these things make it challenging to do things there. Anyway, we generated a knockout. Anytime you take out a gene, so you have a wild type, I took out a gene, and I want to say this gene is responsible, I need another strain, where I put back the gene. What we call as a complementation strain. So if you don't put back the gene and show that the function has, whatever function is lost has come back, then the results are not valid. Means I can take out X, but I may have a phenotype something, but if that phenotype cannot be restored when I put back the gene, it's meaningless. It's some other effect, it may be something else has happened somewhere else, and that is not acceptable. So we do also a complementation strain. So we have 3 strains: wild type, a mutant where original plasmid is put back, original gene is put back in another place, not in the same place. And then what we started doing was doing in vitro experiments. Nothing happened, everything was growing very, very well. You can see that everything is growing as well as the others, which is what the literature already said. But literature also said if we take macrophages and we infect it ex vivo, means you do it like you grow the culture, you take the macrophages, you get the macrophages from mice. You take the mice, you give it certain things, you get a peritoneum is full of macrophages. We take out those macrophages and perform the experiments. When we did that, you can very clearly see that the mutant does not survive as well as the wild type or the complemented strain. Our complementation was spot on. So good that it looks as good as wild type. And then we did the same experiment in vivo. What is in vivo for us? So imagine the in vitro means bacterial culture, ex vivo means cell lines, in vivo means animals. So we took animals, we infected them, and after that, on day 1 we sacrifice the animals, we get the load in their lungs. And then as the time progresses, it establishes infection. You can very clearly see that the mutant is always compromised, and the complemented strain is basically coming back up. So what we have established is the following, that there is a transcription factor that seems to be not essential for growth when we put it outside, but essential for growth when you infect a cell line or when you actually infect a mice models. That is what we have established, right? So, but when a bacteria like macrophages, Mycobacterium enters your body, it is inundated. It's like a war. It is inundated with stresses. What's a stress? You put oxidative radicals on it. You basically you're not going to sit quiet, right? You're going to put oxidative radicals, nitrosative radicals, you put surfactant stress, you put all kinds of stress, including pH is much lower in the phagosomal compartment. So these are kind of known, that these are the things that happen when the pathogen enters our body. So, however, what we do is we take in vitro, this is when you're growing a culture, you can do one at a time. You can make it oxidative stress by adding CHP or H2O2. You can do nitrosative stress by adding those which generate nitrate ions, NO kind of things. We can make it acidic by adding acidic media, things like that. So we keep on doing this. And when we did these experiments, what we found out that in vitro originally it wasn't essential, but when you add either an oxidative stress or a nitrosative stress, in both cases, the pathogen which was a mutant was almost completely dead. You can see this. Whereas other stresses don't work that way. You can see the death happening the moment we put an oxidative stress, right? So why is this important? You're trying to figure out the function of this transcription factor. How do you figure it out? You have to it's essentially doing science and biology is dissecting step by step. It's true in every case, but the way you do it is step by step. So what we identified is the following. In vitro also pathogen dies if we put oxidative or nitrosative stress. And we tried with various oxidative stress, it happens, right? So what we have shown is that either oxidative or nitrosative stress kills the mutant, not the wild type, not the complemented. The question is why, right? So inside the mice or inside our body, what happens is whenever you have interferon gamma, because it's inflammatory response. See, what happens is when you get infected, what you get is an inflammatory response, which are driven by things like cytokines, basically IF and gamma kind of thing. When some of those inflammatory responses happen, what our body does is that we produce oxygen radicals through something called phagocytic oxidase, and also we produce nitrosative radicals through nitric oxide. So all these things contribute to killing the pathogen that is in our body, right? So there are mice where the phagocytic oxidase is missing, or IF and gamma receptor is missing, or nitric oxide can be stopped with adding an inhibitor, right? So what we did is we took mice where IF and gamma, basically we did this the wild type mice where you added IF and gamma from outside into the peritoneal macrophages. You can see you get much more death because you are now producing more oxygen radicals, and nitrosative radicals kill pathogen. But if you take an IF and gamma null mice, you don't get that. Means if you are taking away the inflammatory response, then you are not going to kill the pathogen, and the mutant is going to happily survive, right? Because of what? So we did this with phagocytic oxidase, which I talked about as one of the oxidative radical generation. When I inhibit that, we get some level of survival. We inhibit nitric oxide, we get some level of survival. When we take out both, we get a complete revival. What it tells us that this particular transcription factor is necessary for combating oxidative and nitrosative stress, whether it is outside in the culture or in a cell line experiment or in a animal experiment. I'm not going to go into the details. I'm basically saying even in animal. All these things basically told us that it has some function. What is that function? Right? When you have to find out the function, what you do is omics. So in this case, we did transcriptomics. Basically, we wanted to look into transcription factor, so you expect certain gene expression profiles to change, right? So we took regular culture, S37RV is our control, without and with oxidative stress, and we took the mutant without and with oxidative stress, and we performed RNA sequencing analysis, complete high throughput, right? What we identified is there was hardly any difference between the wild type and mutant in the absence of any stress. There were some 10 genes which are changing, but the moment we add oxidative stress, huge number of changes have happened, falling into many cellular processes, right? What you have to do is validate it, which we did. You can take an H37RV strain, you put oxidative stress, and we took some candidates which go up or go down. You can see very clearly in a QRT, means quantitative real-time PCR experiment, all these RNAs are going up in oxidative stress. In the mutant, they don't go up anymore, right? So it very clearly states the following. I have a transcription factor which is changing the profile of approximately 400 genes in the oxidative stress, right? How? We started looking into one of the candidates which happened to be part of an operon that we talked about. Here is an operon, and this is the gene that was identified in RNA sequencing. These three genes are actually responsible for producing metabolite, or rather amino acid cysteine, right? Cysteine in our body, in bacterial body, can be produced three different ways. One of them is through serine, and the other two pathways involve something else, two other syntheses. This is one of the syntheses where O-phospho-L-serine is the starting point, giving cis-O-cysteine. These three genes are involved in producing cysteine through this pathway. And I'm making it short, okay? So you end up getting cysteine because of these three genes, and it generally is important for antioxidative response of the pathogen, right? So what we basically did is, if that is the case, if this is the case, if I provide cysteine from outside, we should be able to rescue the phenotype that we observed. In fact, that happened, right? When we added cysteine from outside, whether it is in vitro or ex vivo, in both cases we ended up getting a rescue. So it basically told us looks like this gene is actually regulating cysteine biosynthesis through modifying this. We thought that it is because it's auto-regulating and turned out to be wrong. But to cut the story short, we did the other things also, but if you overexpress these 3 genes also, we should get a rescue, because they 3 will contribute to cysteine biosynthesis. We did get a rescue here. So when we overexpress these 3 genes also, we get a rescue, but it was not auto-regulating because levels of the transcription factor do not change. I'm making it a little bit simple here. So what did we identify? We identified that our transcription factor does not auto-regulate, but cysteine gives us the thing back, and also overexpressing 3 genes that are responsible for cysteine biosynthesis give us the phenotype. So we looked into qRT-PCR again to see the expression pattern of these 3 genes in oxidative stress, and all of them go up, right? But when you have a mutant, none of them go up. This is the protein level, this is at the RNA level. So you can see that, for example, AOSR is the transcription factor gone, but you look into this. This is cis-O. It goes up when you add without any in RV, but not in the mutant with oxidative stress, but in the complementary it goes up. So all these things told us the following, that there is something that is happening. Normally there is certain level of cysteine synthesis, but when you add oxidative stress, you are getting these 3 genes overexpressing. Means there is another promoter. Okay, there is a normal promoter, but when you have oxidative stress, there is another promoter. So just again, we tried complementing with only one of them. This is not important. So what we did, we had a hypothesis, there is an alternate promoter. How do you prove you have an alternate promoter? In biology, all that you do is you take those 500 bases in that and attach it to a reporter. What is a reporter? It can be GFP, it can be luciferase, it can be anything. We attached it to luciferase. Okay, here is a scenario. This is the reporter, this part of the protein, in front of the system, this part of the gene which is attached to a luciferase. You can very clearly see that it goes up 100-fold in wild type strain. But basically saying that there is an oxidative responsive promoter there. When you put it into the mutant, AOSR mutant, it doesn't go up. So we identified an alternate promoter, novel alternate promoter, and we basically showed that, I'll just cut down this bit a bit. We identified an alternate promoter, and then we performed what we call as an interactome experiment, which is nothing more than pull down your protein, whoever comes along, who are the neighbors or the interacting partners. We got one of the alternate sigma factors as a pull down partner. Sigma factors, as I said, is important for transcription because they are involved in recruitment of RNA polymerase to the promoter. There is a housekeeping sigma factor that takes care of 2000 genes, that is sigma A, and there are other sigma factors. We basically identified sigma H, which is an alternate sigma factor, to be an interacting partner. So what does it mean? Basically it means that we showed that it actually interacts both in vitro and in vivo. I'm not going to this. And basically what happened is that we basically showed in the previous experiment that sigma H interacts with AOSR only in oxidative stress. Normally it doesn't. And they together bind to an alternate promoter. How do you show that? You do something called gel shift assays. You label a small fragment of the DNA, you incubate with the protein and see if the DNA shifts. It's called gel shift assay. You can see very clearly shifting. And what we then showed is that you add sigma H along with the transcription factor, the shift is more prominent, indicating that together they bind much more robustly to the promoter than independently. So all these things more or less told us that there seems to be a particular way in which the whole thing is working, and this is, I will not go into it. We showed it biochemically in many other ways. So one of the things that is left, basically we demonstrate till now, is transcription factor along with an alternate sigma factor binds to an alternate promoter in oxidative stress, increases cysteine. Is it true? you have to do something called metabolomics. You have to measure the metabolites that are produced at the end of this kind of an hypothesis, right? So we did metabolomics. We can very clearly see that the thiol levels are higher in the wild type and in the complemented strain, but not in the mutant. MSH, mycothiol, is also higher in the wild type, complemented, but not in the mutant. Ergothioneine, higher in the wild type, mutant, but not in the wild type and complemented, not in the mutant. We cannot measure cysteine because cysteine is very sparsely present in bacteria. It keeps on moving to other metabolites. So we clearly established that it is controlling metabolism, specifically cysteine mediated metabolism, right? So that's the story where, I would stop today because I really feel that, going any further will take more time, and also I don't want to complicate the matters. Anyway, so what we did is the following. We've identified that sigma H in oxidative stress becomes free, interacts with AOSR, forming a complex. This complex is then recruited to an alternate promoter, getting you higher levels of transcription of these three genes, which in turn produces higher level of cysteine, and that leads to many of the other metabolites going up. This was the story that we actually came up with. And so what I'm basically trying to convey here is that we are dealing with a very intelligent pathogen. It finds very, very small ways. In this particular mechanism is not even conserved in non-pathogenic soil bacteria called smegmatis. So even within mycobacterial species, this mechanism is not conserved. This seems to be specific for the pathogenic bacteria, because pathogenic bacteria is the one that faces this niche. So it has mechanisms and mechanisms and mechanisms to survive in the host. All I've given you is given one of them. And if Dheeraj were to talk, he will tell you many other things about how it manipulates the host. It manipulates what kind of cytokines are secreted, and it is capable of surviving in neutrophils. It is capable of surviving in mesenchymal stem cells. So it is something, it is somehow evades all that we throw at it from our immune response and gets into corners which you can't really destroy and manipulates your thing. For example, we in another independent work showed that it causes DNA damage not to kill us, host DNA damage not to kill us, but to make sure that the apoptosis is inhibited. So things like that. It's a pathogen with 150 mechanisms that is kind of, it's evolved to make sure that it survives in our body. And that is why from mummies to us, it continues to be there. And luckily there are antibiotics to take care of it. All that we have to do is make sure that we use those antibiotics properly and not use them in a bad way. In the sense, what is a bad way is non-compliance, so that we don't get resistant bacteria. So TB is a treatable disease, but a painfully difficult one. Thank you.
Questions?
Here.
Hi, fascinating talk. So I was just wondering, you mentioned that the bacterium has so many different strategies that has evolved. Is it possible by looking at, this, early historic samples of tuberculosis bacteria? I don't know whether you can actually examine them at that level of detail, but is it possible to see whether some of the survival strategies are actually later
evolved or not? Very interesting question, but I wish we have that particular insights easy to get. I'll tell you the reason. Okay, you have to not only get pathogen. See, there are two levels. One is genetic, which is genome. In genome level variations, you can pull out an old one and you can look at a new one and you can make comparisons if you get sequence from mummies to a current one. But if you start talking about what he said a while ago, transcriptional manipulations or epigenetic manipulations, things will be a little bit different. And it is very difficult to capitulate those differences from that time to now. So at this point of time, if we can do a good job of looking at the differences, at the heterogeneity rather, at both at a genetic level, epigenetic level, and at the transcriptome level of, say, pulmonary tuberculosis versus extrapulmonary tuberculosis, drug resistant versus regular one. If these kind of things are done in itself, we will get much more insights. But evolutionarily, it's an interesting pathogen that seems to have co-evolved with us. But it will be, the valid question, difficult to address.
Are there other animals who also suffer from tuberculosis?
Yes.
And is it possible for cross-infection between human and...
Currently, the vaccine that is available is BCG, right? It's Bovis BCG essentially. It has come from Bovis infections, right? Bovis is cows. So you have tuberculosis in animals. So, but they are not exactly the same as the tuberculosis that gets to us. But they can cross-contaminate, cross-infect. It's true, they have the capability, but the evolution is also more towards its host, okay? When ultimately COVID jumped from, say, animal to us, right, it completely evolved in us and became a much more robust strain, like in case of what you call as Delta. And at a later point of time, it became even more interesting by finding more mutations. So evolutions continue to happen in the host, but cross jumps do happen. Can be really infected with BCG. There is one which causes Mycobacterium, TB in fish, marinum, M. marinum. So you have Mycobacterium going into various hosts. And yeah, when immunocompromised, life is anyway tough.
That the environment, the human environment in which TB has evolved, would it make it easier or harder for it to jump to other species and vice versa? Like, is that a meaningful question?
Yes, it is. But I believe that that's what he said. If you are immunocompromised, a species jump is relatively easier, because then it is basically getting into your thing and you really don't have enough mechanisms to eliminate it. But if you are a healthy individual, because species jumping organism is not evolved to survive in you, you can eliminate it much easier. So these things happen. There is even a hypothesis for Omicron, right? It is actually perpetuated in an immunocompromised individual accumulating as many mutations as it did. And this is a hypothesis only. But this is the way in which evolution occurs, right? But it doesn't mutate the rate anywhere closer to the rate the viruses do. So we don't get, mutated tuberculosis just like that.
Any other question?
So you spoke about how this sigma H factor is like it binds to this upon only oxidative stress. So how does sigma H sense oxidative stress?
So I did not get into it. So sigma factors, there are two kinds. Housekeeping, of course. Then there's something called ECF sigma factors. These are the sigma factors that are kept in check by binding with an anti-sigma factor. So normally they are in a complex. So the sigma factor is not available. When oxidative stress happens, the anti-sigma factor either gets phosphorylated or the way its conformation changes because of sometimes some of these residues, cysteine getting SS instead of SH, two cysteines coming and becoming a disulfide bond. Multiple things happen. So because of that, now the sigma factor becomes free.
So the partner, the inhibiting,
it exchanges the partners.
So the partner has two proximal.
In this case, there is a phosphorylation is known, in case of the sigma H anti-sigma factor.
The phosphorylation is done by a
one of the
kinase which
kinases.
senses oxidative stress?
Part is not known, which kinase does it and why it does it only in oxidative stress is not known. But phosphorylation is known to be a mechanism where this dissociation happens is known. And there are some other anti-sigma factors where they get degraded. So releasing the sigma factor. So there are a lot of work that needs to be done at a basic biology level to understand how various processes work, including transcription, that is including sigma factors. There are 13 sigma factors, and if you were to ask me which sigma factor is involved in when and all those things, we are addressing that question going forward. But it's something that requires a lot of basic work.
There will be a huge interactome to dig
chip
through.
sequencing,
Yeah.
RNA sequencing, interactome, all three, and in multiple conditions.
Yes. Thank you.
No, none of them are essential other than one, sigma A.
Sir, your work is mostly about this oxidative stress, what happens during oxidative stress. So, is this happening in the body?
Yes, in the body, there is oxidative stress that we put in. Whenever you have your body macrophages, so we have what you call inflammatory response, right? Inflammatory response means the moment IF and gamma kind of things bind to its receptor, you end up actually getting oxidative radicals generated on the phagosomal membrane and also getting nitrosative radicals generated. So this is how we actually combat. We also combat and limit the survival of the pathogen.
So, is this happening in all the individuals or specifically in some?
It will happen in all the individuals. If you are relatively more compromised, your ability to do most of these things is not as robust.
Thanks.
Immunity can be blessing and also a problem. Sepsis, it is the problem, right?
Any other questions? So I had a nice question. So I should think of this alternate promoter as like these are conditional operons?
Yes, it is a conditional. Yeah, you can say so.
Oh, thank you.
That's a different system for the pathogen. Normally, pathogen doesn't let these three genes get separately expressed, but under the oxidative stress, it allows them to be expressed separately.
So your hypothesis is that we will see more conditional operons in pathogens? Pathogenic bacteria?
Possibly. We have to look for it. It's not looked for, but this is not the only mechanism. I believe there are more. We elucidated. It's kind of known.
Any other questions?
Because you have alternate sigma factors, they are going to in itself sufficient, but you also have transcription factor. Together, they'll make enough conditional promoters.
Questions? If not, then let's thank Vinay again for this wonderful talk. There's high tea outside at 4, so I'm assuming there will be high tea. So you're all welcome to join. Thank you.